Hello and welcome everybody to our webinar by Bioreperia. I see that some of you have logged in. Well, you're all very welcome. This is our fourth event, our fourth webinar for this semester. And the webinar series is called Something Smells Fishy, Removing Doubts, Zebrafish, an exceptional translational in vivo tool for drug development and personalized medicine and cancer. And I know there's been some issues, I think, with the timing, but I'm very happy to see that you have found your way here. And today, Lasse will be presenting. He's our CTO here at Bioreperia and also the founder of the company. Bioreperia was founded in 2015 as a spin-off from Linköping University, and our main focus are working with zebrafish in the field of oncology. So I will give the floor and the word to Lasse to continue this webinar. And if you have any questions anytime throughout the talk, please write them in the question section, and we will address them after the presentation by Lasse. The floor is yours, Lasse.
Great, thank you. Thanks. Yes, welcome to episode number 4. In this episode, I will be talking about immune oncology and how to understand and model the complexity of human cancer immune cell interactions using the ZTX models. So first, a little briefly about the drug discovery and development process. It's, of course, consisting of different steps. In this talk, we are mainly focusing on the preclinical or translational steps. Here, step number 2, before a drug is coming into clinical trials. This could also, of course, be relevant for basic research, but the ZTX models are often used once the most preliminary studies have been completed. So I would put the topic of this talk under category number 2 here. The ZTX models are based on implanting tumor material, either cell lines from in vitro cultures, or a combination of different cell lines, as I will show you later. Alternatively, cells isolated from tissues, implanting these cell suspensions into the perivitelline space, which is a subcutaneous compartment of the zebrafish larvae, and then analyzing the effects of either small molecules or antibodies in this system. That will, of course, lead to a report detailing the anti-tumor efficacy and also anti-metastatic efficacy of these compounds on this homogeneous or heterogeneous tumor that is being studied. And the results are ready already within an assay time of 3 days. So it's a very rapid in vivo model system for analyzing anti-cancer efficacy and metastasis. It looks a little bit like this. A primary tumor is usually maintained at a certain size. Under controlled conditions, you can often see that they start locally invade the tissues after 2 to 3 days. But commonly, you do not see much growth because it's only a 3-day period. And the temperature is approximately 1 degree lower than the physiological temperature of the tumors, 36 degrees, meaning that you don't see a rapid growth within these 3 days of the tumors. What you do see, however, is if an efficacious drug is added to the water or injected into the fish, you can see how it kills the tumor cells. In this case, the drug will only kill the tumor cells. It will not inhibit the local invasion, as you can appreciate from this image at day 3. So this is the type of results that come from the ZTX analysis when it comes to the primary tumor. I mentioned that the tumors can be established from different cell sources. For example, a tumor cell line, a CDX model, cell line derived xenograft model, can be combined with other elements of the tumor microenvironment, for example, non-malignant infiltrating stroma cells or immune cells, to reconstitute the elements of the tumor and the tumor microenvironment that you're interested in studying. In this particular talk, we will be focusing mainly on reconstituting the immune cell compartment, but I will also show you some data on the effect of adding certain types of stroma cells to the system. In addition, we are doing in BioReperia quite a lot of studies using complex mixtures that are derived directly from patient tissues. So tumor biopsies taken from the patients are maintained with the cells that they consists of and recapitulated in the zebrafish subcutaneous environment. So in this case, we do not have control over exactly what cell types or how many of each cell type, but we simply reconstitute the tumor found in the patients. I will also show you some data related to this. However, the clinical aspects of the ZTX model system will be dealt with more in depth in the next and fifth episode of this webinar series. Tumors can be implanted in different sites in the zebrafish embryo or larvae, as it's called once it's hatched. In this talk, we are mainly focusing on subcutaneous implantation of tumors. This is the way that solid cancers are established in the larvae, and it will allow both visualization of the primary tumor and local invasion, and effects, of course, on regressing primary tumors, as well as the metastatic capabilities of these tumor cells and the effects on inhibiting metastasis. Certain types of tumors, mainly soft tissue cancers, can be reestablished in the yolk sac. It's a very soft tissue environment that might be more similar to their local environment for some soft tissue cancers. This is, however, not a good place to implant solid tumors. So we recommend that solid tumors are always implanted subcutaneously, if not in the organ in which they are derived. Blood cancers, however, are often studied following intravenous injection. Also, if you're focusing on the metastatic process, you can also, of course, implant the solid tumors intravenously, and this will lead to a systemic establishment of the cancer in the entire larvae. And of course, the progression of the metastatic tumor cells or the blood cancer in the fish can then be evaluated over time in the presence or absence of drugs. Here's an example of what it can look like when a tumor is reconstituted from both tumor cells and neutrophils, tumor cells shown in red and neutrophils in blue, and it demonstrates how the tumor also attracts host zebrafish blood vessels shown in green, and really lead to the establishment of a heterogeneous tumor consisting of different cell types that are also locally invading the environment and the tissue next to the implantation site. The recruitment of blood vessels into the tumor is also key for the delivery of drugs that are administered through intravenous injection, or delivery of T cells administered through intravenous injection, and therefore recapitulate the clinically relevant administration route of the treatments given to the patients. This is important as such an administration route can be used to identify whether T cells can home in to the tumor with or without certain treatments. I'm going to show you some examples of that. And of course also how inflammatory cells or other stromal cells, I will show you an example with fibroblasts later, may modulate the immune oncology response, or the response of the T cells towards the tumor cells. In addition to the primary tumor, you can use the zebrafish model to evaluate metastatic spread. That's often evaluated in this caudal hematopoietic plexus, which is similar to the bone marrow in patients. This is the hematopoietic region of the zebrafish larvae. And you can see in this image that it's also the place where the tumor cells preferentially metastasize. You don't see too many tumor cells metastasizing to the muscle, and often also not to the brain. Of course, that depends a little bit on the tumor cell line used. But in many cases, you will see metastasis in this caudal hematopoietic plexus, and this can be visualized in high cellular resolution. And of course, you can also see whether your T cells will recognize these micrometastatic nodules and kill these tumor cells at the metastatic niche. And I will also show you examples of this later. When it comes to understanding the innate immunity and how it contributes to both the primary tumor development and metastasis, there's been a number of publications in the recent years on this topic, mixing tumor cells with macrophages, neutrophils, fibroblasts, adipocytes, but also with T cells, and I will expand a little bit on this later on. First, I will show you a few examples of how the effects of macrophages and neutrophils and also fibroblasts have, in the tumor microenvironment, have contributed to the understanding of the role of these cells and how pathways, pro-tumorigenic pathways elicited by these aspects of the tumor microenvironment can be targeted therapeutically. In one example, Liu et al. Demonstrated that fibroblasts can recapitulate the pro-tumorigenic aspects of carcinoma-associated fibroblasts, or CAFs, excuse me, sorry, by mixing different types of tumor cells. For example, in this case, hepatocellular carcinoma tumor cells with CAFs derived from either a prostate cancer or a colorectal cancer. You will see that these CAFs, they lead to markedly increased local invasion, but also distal dissemination to this hematopoietic caudal plexus compared to the hepatocellular carcinoma cells themselves that were moderately invasive in the local environment, but not nearly as much as seen when they were combined with the CAFs. This was specific for the CAFs, as normal fibroblasts did not elicit a pro-metastatic phenotype of these hepatocellular carcinoma cells. A similar study, but instead using tumor-associated macrophages, or TAMs, has been conducted by Wang et al. Also here, you could see that TAMs isolated from patient cancers. 3 patients were studied here. 1 patient with non-metastatic endometrial cancer, 1 patient with metastatic endometrial cancer, and 1 patient with metastatic colon cancer. We took a small tumor biopsy from these patients, isolated the TAMs, and mixed them with otherwise non-invasive tumor cells. Also, fibrosarcoma cell line used in this study here. Mixing these non-invasive cells with these TAMs, we saw that TAMs from the metastasis negative patient also was not able to elicit an invasive phenotype, metastatic phenotype, in the zebrafish model, whereas the TAMs from metastasis positive patients changed the metastatic phenotype of these otherwise non-invasive tumor cells into a highly invasive tumor, both locally and also a tumor that seeded distal metastasis to a much higher extent than without TAMs or with TAMs from a metastasis negative patient. So this demonstrates that these critical aspects of the tumor microenvironment can really turn tumors from a non-invasive to an invasive phenotype. Similar study was done with neutrophils, showing that estrogen receptor positive breast cancer cell line here that is not metastatic. You see very few metastasized cells, and even if you add estrogen, the tumor did not become metastatic. When you added neutrophils, however, you could see a significant increase in metastasis, so the neutrophils alone could confer invasive phenotypes on these MCF7 breast cancer cells. However, if you both added estrogen and neutrophils, you could see really a synergistic increase in metastasis, the estrogen-stimulated MCF7 cells with neutrophils metastasized to an incredible extent, both compared to neutrophil mixed cell combinations alone or estrogen-treated cancers alone. So this was really a remarkable example of how different aspects of the tumor microenvironment, in this case estrogen and neutrophils, can synergize in mediating invasive phenotypes of these tumors. Now, moving from innate immune cells and fibroblasts towards the real topic of this talk, namely the T cells and immune oncology treatments, there has also been a number of papers recently. You will notice here that most of these papers are not more than 1 or 2 years old, demonstrating the use of zebrafish models to analyze anti-cancer efficacy of CAR T cells or tumor-infiltrating lymphocytes. And these are the 4 papers in this area at the moment, at least when it comes to studying this in zebrafish larvae. There are a few more papers studying this in adult zebrafish, but in zebrafish larvae, these are the 4 studies. So you can look into these for more details, but I will also show you a few examples from each of these papers in the coming slides. First out was Pascual et al. That in early 2020 published a study showing that CAR T cells potently killed NALM6 lymphoma cells. You can see the green lymphoma cells present here in the caudal hematopoietic plexus at 2 hours post injection, but they were all eradicated when CD19 enhanced CAR cells or CAR cells that were genetically manipulated to activate the T cell receptor signaling when engaging CD19 were added to the system. Having no CAR cells, you could see that the cells would not spontaneously regress. They would indeed establish microtumors in this hematopoietic caudal plexus. So the regression of the tumor 24 hours after adding the CAR T cells was CAR T cell specific. A similar study was done by He et al. In the same year, demonstrating instead when using a control situation with the peripheral blood mononuclear cells, PBMCs, they were not able to regress the green melanoma cells here when they were co-injected in the subcutaneous base. However, if TILs that were isolated from the same patient that contributed the melanoma cells, when they were injected together with the melanoma cells, these TILs were really highly effective at killing the melanoma cells, thereby demonstrating that they do have activity on their own. So if these TILs were not able to kill the melanoma cells in the patient, this might be because of other aspects of the tumor microenvironment that was not recapitulated simply by reconstituting the tumor from in vitro cultures of the melanoma cells themselves and of the TILs themselves. It was also shown in the same paper by He et al. That a similar system would demonstrate the efficacy of CAR T cells, again anti-CD19 CAR T cells against Raji lymphoma cells. When mixing Raji and CAR T cells together and implanting them in the or injecting them IV, you can see that the Raji and CAR T cells, they co-localize in the hematopoietic caudal plexus, and 24 hours after injection, all of the Raji cells are killed by the CAR T cells. And this was not the case if instead of an anti-CD19 CAR T cell line, a control CAR T cell line that instead was expressing green fluorescent protein was used. So these control CAR T cells could not kill the Raji cells, but it required the CD19 modification. Also, He et al. Demonstrated that this ability of the tumor infiltrating lymphocytes to kill the melanoma cells that were clear in the zebrafish tumor system when only these two cell lines were used, but which was not seen in the patient. This tumor killing ability of the TILs could be inhibited by adding cancer-associated fibroblasts from the same patient. So now when reconstituting yet another aspect of the tumor microenvironment, namely the carcinoma-associated fibroblasts, that led to a complete annihilation of the efficacy of the TILs to kill the melanoma cells, demonstrating that the CAFs have immunosuppressive functions, which others have demonstrated as well, but this is shown in a very visual manner here. The green melanoma cells completely dead when adding only TILs and melanoma cells. However, when adding the CAFs, the green melanoma cells are retained and have the same volume as upon implantation. And this was further elaborated not for CAFs but for macrophages in a subsequent study by the same group, Wu et al., that demonstrated that macrophages that are not stimulated with IL-33, so in naive macrophages, they were not able to impair the TIL cell mediated killing of the melanoma cells. You can see the melanomas become much smaller when adding the TILs. However, if the same macrophages was pre-treated with IL-33, which these authors found led to an induction of MMP9 by the macrophages that in turn cut MHC class 1 from the monocytes, meaning that the TILs could not recognize the melanoma cells any longer. Now the TILs could also not kill the melanoma cells. The melanomas remained at the same volume 24 hours after adding the TILs. No significant difference in size. So in this case, it was a macrophage-mediated mechanism that incurred immune resistance to these melanomas, demonstrating again that even molecular aspects of the interactions between T cells and tumor cells can really be studied in cellular detail in vivo and in only 24 hours using the zebrafish model system. Now, at Bioreperia, we have done 3 studies taking these aspects one step further. In one study, together with a pharma company from Stockholm, we have looked at tumor-trained lymphocytes from a patient, that is T cells isolated from a cancer patient and trained with tumor neoantigens to be activated against the tumor. Then taking tumor cells from these patients and these personalized tumor-trained lymphocytes and co-injecting these cells, we saw that the group with the tumor-trained lymphocytes really had a marked regression of the tumor volume, meaning that these PTTLs were effective at killing the tumors. This was the case for a patient with urinary bladder cancer, and also demonstrating the translational power of the zebrafish model. So this was indeed a patient that donated these both T cells and tumor tissues to us for this study. No expansion of neither the tumor cells or no expansion of the tumor cells had been done. The PTCLs were of course expanded when they were activated with the tumor neoantigens. In a study done with a pharma company in Israel, looking at the efficacy of checkpoint inhibitors, we studied melanoma and saw that melanomas that grew a little bit from day 0 to day 3, here shown in blue color, they were not impaired when adding checkpoint inhibitors alone. So checkpoint inhibitors did not lead to a regression of the of the melanoma on their own. Adding TILs from the same patient that donated the melanoma cells, of course, led to a slight reduction of the melanoma size, as you can see, comparing the second and third row here, and as you can see, also comparing the second and third graph. But if you added both TILs and checkpoint inhibitor anti-PD-1 treatment, you could see a really dramatic reduction of the primary tumor volume, demonstrating that these semi-effective TILs could really become highly effective and have augmented tumor killing capacity when adding checkpoint inhibitors to this system. Again, demonstrating that even drugs with a complex mechanism of action, such as checkpoint inhibitors, their efficacy can be clearly demonstrated in the zebrafish tumor xenograft models within only 3 days. Finally, with a big pharma company from Switzerland, we found that a drug that they are developing that is a bispecific antibody with one arm binding tumor cells and with the other binding CD8 positive T cells. Adding this bispecific antibody with tumor cells shown in red and T cells shown in blue, you could really increase the co-localization of these two tumor cell types, ensuring that the cytotoxic T cells are preferentially binding to tumor cells. This, in turn, led to a dramatic reduction of melanomas. You could see that the melanomas were greatly reduced when adding this bispecific antibody at day 3, compared to if the bispecific antibody was not added. But this approach was not specific for colorectal cancer, HT29, suggesting that there is an immune modulatory or immune inhibitory signal present in these tumor cells. And that, of course, remains to be found what that signal might be, but it was quite clear that the T cells definitely co-localized with the tumor cells, but why they were not able to kill the tumor cells when they were very effective at killing the melanoma cells, that is currently not well understood. So we are looking into that further. So in conclusion, I hope that I have convinced you in this webinar that the ZTX models are really accurately recapitulating relevant aspects of immune oncology and can be used to study the efficacy of drugs in this space, including autologous T cells, CAR T cells, checkpoint inhibitors, and bispecific antibodies that bind T cells and tumor cells. With this, I will take your questions, but also first I will give you a little teaser that in the next webinar in January, we are going to show you more data on how the ZTX models can be used for personalized medicine and also for personalized immune oncology, looking at the role in predicting treatment outcomes in the patient. So that will be the next webinar here that's scheduled for the 12th of January at 12. So at the same time as this webinar, 12 noon Central European time, and looking forward to seeing you there. And I would like to take your questions.
Thank you so much, Lasse, for a very nice presentation. We have a few questions coming in. We actually have 2 in regards to if the zebrafish own immune cells or immune system could affect the immune oncology setup that you have described.
Yes, that's a great question, and indeed there has been at least one report that I know of showing that that the neutrophils and macrophages that are present at these stages in the zebrafish larvae may indeed exert anti-tumor effects and increase the innate rejection of tumors that are implanted. In some cases, other tumors will not to the same extent activate this pathway. and may not spontaneously regress to the same extent. And this spontaneous regression that was seen for some tumors seemed to be dependent on the zebrafish's own neutrophils and macrophages. So indeed, they do seem to contribute to the tumor microenvironment and can sometimes have certain effects in this microenvironment. Yes.
Thank you, Lasse. We also have another question in regards to CAR T cells and the possibility of using the model for screening different lines of CAR T cells as a cell therapy in this setup.
Yes, the model is definitely well suited for that. So we have done something similar for the PTTLs. So that's not genetically manipulated T cells as the CAR T cells are. But still, we have demonstrated that different populations of T cells may have different anti-tumor efficacy, and that can be identified when using the ZTX models. So similar to looking at different drugs, whether one drug has efficacy and another doesn't, then the model can also be used in a very similar way to demonstrate whether one type of CAR T cells may have better efficacy compared to another type of CAR T cells in an individualized fashion, of course.
Great, thank you. I think you mentioned this a little bit, and maybe it's not really for this scope, but I will ask it anyway, since we have a question in regards to evaluation of anti-angiogenesis compounds, perhaps then in combination with immuno-oncology setups. It's not really stated.
Yes.
Do you have any comments, or could you elaborate a little bit on that?
Yes, I mean, it's a great concept to bring up. So we have demonstrated that that the ZTX models indeed can be used to study anti-angiogenic treatments. This was demonstrated in some of the first studies using this model system by Marco Presta's group in Italy and by Yi Haikau's group in Stockholm. And they found that different types of anti-angiogenic drugs exerted similar effects in this zebrafish tumor model. In other studies, it has been shown that anti-angiogenic drugs, when used in the right concentration, so not too high concentration, but at a concentration that normalizes instead of eradicates the tumor vasculature, may indeed lead to increased infiltration of lymphocytes into the tumor. These normalized vessels have improved perfusion and also improved diapedesis of the lymphocytes. Thereby, anti-angiogenic drugs at these normalizing concentrations can synergize with immune-oncology treatments, giving a better effect of these treatments. There are other mechanisms that can be considered there as well. For example, normalizing blood vessels have by some researchers been suggested to improve tumor hypoxia, and tumor hypoxia in turn is one mechanism, one immune suppressive mechanism that has been suggested in the tumor microenvironment as well. And these aspects of how hypoxia influence the anti-cancer abilities of infiltrating lymphocytes is also something that can be studied in the zebrafish system. So many of these questions, if not all, can indeed be studied in the fish system. I might want to say that of course not everything is perfect, and also not the ZTX models. So one aspect that is difficult to study is vaccinations. The zebrafish larvae do not have their own B and T cells and do not have a functioning thymus or lymph nodes or other things that are relevant for vaccinations. So anti-tumor vaccinations is probably not an area that can be studied in this system, at least not at present. Perhaps tools are developed to overcome this problem, but not now.
Great, thank you, Lasse. And perhaps also in relation to this question, we have a question in regards to infiltration of lymphocytes into the primary tumor, and whether or not that can be evaluated in the ZTX model. and how it might be affected in the presence of immune checkpoint inhibitors?
Yes, so indeed, we have demonstrated, and I showed you some images of how tumor infiltrating lymphocytes indeed can be seen both within the primary tumor and associated with the tumor cells in the metastatic niches. So it is very much possible to study and quantify the amount of infiltrating lymphocytes and whether this can be augmented. You can have more lymphocytes infiltrating the tumor with certain treatments. With checkpoint inhibitors, I'm not sure if that would lead to an augmented infiltration into the primary tumor. It would certainly lead to a more efficacious tumor killing interaction between the tumor cells and the T cells that have infiltrated in the tumor. But perhaps the main mechanism of action is not the infiltration itself, as far as I understand the mechanistic aspects of checkpoint inhibitors, at least.
Thank you so much, Lasse, and thank you very much for your presentation. Just to wrap it up, I have one question, and many of the articles that you presented they're fairly recently published. So it feels like using the zebrafish as a model for IO and also for TME is fairly new still. What do you feel about the future for this model in this specific areas?
Yes, no, indeed, this is a very new area of research, and I'm sure that there will be a lot more preclinical studies coming out using the zebrafish for understanding mechanisms related to resistance to immune oncology treatments, for example. So this is, of course, something that is coming up. It is becoming clear that a large number of patients are resistant or develop resistance to immune oncology treatments, and the mechanisms underlying this are not well understood. So that's something that can be studied, as I showed for the CAFs and for the IL-33 stimulated macrophages. But there are most likely other mechanisms that play a role as well, and those can be revealed using the ZTX models. However, what I'm more excited about is these personalized medicine aspects, that you can really identify which patients would benefit from immune oncology treatments, and perhaps also which type of immune oncology treatment. Is it the autologous T cells or the checkpoint inhibitors, or perhaps a combination of the two, thereby really improving the treatment outcome for the patients when they are given these treatments. We want to instead of just understanding why they are not responding, that's of course nice, but it would be even better to make sure that they're getting treatments they respond to improve their survival and quality of life. So that's something that I hope the zebrafish tumor xenograft models, ZTX models, can help to accomplish. We will of course elaborate more on that in the next webinar.
Great. Thank you so much for that, Lasse. And that wraps up this webinar from BioReperia. I hope you enjoyed it. It will be available to view later as well. I will share that with you. And please join us for our next webinar on the 12th of January, also at 12 at noon. Thank you very much. Have a nice day.
Thank you. Bye.
Bye.